I want to thank you for inviting me. I think I was complaining to my son that I don't like giving afternoon talks when I teach. I like teaching in the morning, and then I like to give lectures in the morning. I like to do seminars at noon or earlier. And then he pointed out that it's actually 4:30 in the morning in Los Angeles, so this will be the earliest talk that I've ever given. The other thing I wanted to say is that in Los Angeles, where they make movies, they tell you never to be in if you're an actor, never be in a movie with a child or a pet, right? For obvious reasons. And if you're at a science symposium, never follow Amir, because he's literally made the blind see. There's an interesting thing I was thinking about when he was talking. And since I like to go to symposia now or conferences where it's not just more of the same, like here's another talk, another talk, another talk, he got me thinking, where is he? Where is there you are. Okay. So the difference in size between a lesion-cephalic animal like the mouse and the human, the different size of the volume of the cortex, of the cerebral cortex, is about 1,000-fold. Increase in size. What's the difference in thickness between a mouse cortex, gray matter, and human? Does anybody know?
5-fold.
How many fold? 5-fold? Anybody else?
1.5.
Exactly, 1.5. So there's only about a 50% difference. Give him a hand. Here, wait a minute. Whatever. I don't have a lot to say with my data, so I might as well do this. So 1.5 fold. And which layers or layers are responsible for that increase in thickness? Almost exclusively. Layer 2, 3, right? So the expansion in primate cortex is in the tangential domain. All these association areas that we assume we understand are association areas and therefore are multimodal, multisensory. We have really very little understanding about, except that we map them in imaging studies, but don't really understand how they develop, how they arrive in terms of their level of complexity and also their capacity for what you've just shown. So I think it's brilliant. And it fits, well, that's okay. He also paid me to say that. But it fits with this idea that the plan, the developmental plan for building the cortex in terms of the radial domain is set in evolution. It's highly conserved in terms of laminar organization, but really what changes are all the things that you talked about in terms of representation in the tangential domain, building areas that simply are not represented in other species. So anyway, I want to start with this video. Let's see if it works.
You can think of child development as a scale that has two sides. A child's scale is placed in a community and has spaces on either side where experiences and environmental influences stack up over the course of development. One side gets stacked with negative factors like stress, violence, and neglect, while the other side gets loaded with positive factors like supportive relationships, skill-building opportunities, and community resources. In the same way that the weight on a teeter-totter affects the direction it tips, the factors that a child is exposed to affect how they turn out, the outcomes of their development. But the way the scale tips and the outcomes a child experiences are also a function of the position of the scale's fulcrum, which is like the genetic makeup of the child. The position of the fulcrum affects how the scale responds to factors that get stacked on either side and how easily it tips. We know that fulcrums start in different places for different children, and that this genetic starting point is an important factor in how a child responds to experiences and how they turn out. We also know that the position of the fulcrum isn't fixed. It's a sliding set point. Over time, the things that load and tip the scale can actually shift the position of the fulcrum. There are points in human development when this fulcrum is easier to shift. During these shiftable periods, our biology can be changed, making us more or less able to withstand negative weight that gets stacked on the scale. One of these shiftable periods is early childhood, and another is adolescence. That's why it's so important to make sure that children have positive experiences, especially early on, and to continue positive supports throughout development. By providing positive experiences during these sensitive periods of development and offloading negative weight, we can shift the fulcrum, making children more able to bear the weight of negative experiences later on, making them more resilient. So resilience isn't just about individual children. It's about the environments and experiences that get stacked on the resilience scale. Resilient societies are those that figure out how to stack more positives than negatives onto the children who will become workers and citizens.
So that video essentially summarized, I can stop now because the focus is what are the variables that influence vulnerability and resilience during development. And there's all that we know, and we know a little bit, is summarized in that 1 minute, 30-second video. But one of the most important concepts that's articulated in that video, and that video was made to show to non-scientists, to policymakers and others, to get them to understand that investing early makes a whole lot more sense than waiting. And most of the dollars that are invested that have to do with human beings are invested in the last 25% of life, not the first 25% of life. So it's a simple message, but Amir, one of the criticisms I would have of your great talk is that you used nature versus nurture. And genes are actually not built to not respond to the environment. They're actually built to respond to the environment. So genetics is not superglue, right? And so that's very important. That fulcrum that defines that helps to define vulnerability and resilience actually moves based on experiences that an individual has, irrespective of all the animal model studies that we do and that I'm going to describe. This paper just came out. It's a study that essentially replicated the adverse childhood experience study that Kaiser did in the 1990s. What was interesting about that experience, that study that the Kaiser health system did, is that they realized that in their obesity clinic, they identified a larger than expected number of individuals in that obesity health clinic that were reporting early adversity, which included neglect, abuse, witnessing violence, mental health issues with their primary caregiver, et cetera, and you've heard some of those already today. If you look at this new study, in the United States, about 62% of those who were in this study, and it's a very, very large study, 215,000, had at least one early adversity experience. 25% had 3 or more, so that's 1 in 4, are in the range where you start seeing increased risk for all sorts of problems that I'll get into in a little bit. So this is a common problem, and a problem that we understand very little about. Now, what was curious about the Kaiser study is they developed a questionnaire, the ACE questionnaire, that has 11 items on it, and it's been validated. It's been used in a number of different studies, and it's simply not used clinically, right? So the Kaiser system that funded this research is a health system in California. They did this research, they generated the data, and then they just basically said, okay, here are these findings. 1 in 4 have 3 or more adverse childhood experiences and don't use it clinically at all. So the conundrum we have is how are we going to scale what we do in terms of research? How are we going to understand, how we're going to take this information and translate it into best practices? And one of the biggest problems is identifying those who are at risk and those who have resilience to these kinds of experiences. Okay, so there's that. Okay, so what I'm going to talk about, I'll talk about some of the variables that we think are involved in creating vulnerability. Not much about resilience, except some work that we're just starting to do in terms of doing genetics in animal models. So there are heritable factors that influence the heterogeneity of complex behavior. I'll just mention briefly some of the work that's already been published in 2017 on the genetics that may be responsible for the wide range, the heterogeneity of affiliative social behavior. And it follows on Epstein's work showing that in twin studies, heritability for some pro-social behaviors is very high, 0.5, 0.6. That's high in terms of heritability. Heritable factors vary the response to experience due to, sensitive period changes, not critical periods. There's no such thing as a critical period. But you can vary the timing of that, and yet the experience is the same, and I'll just talk briefly about that. Vary the response to experience due to differences in environmental context. The same genetic variation actually can be responsible for two different responses depending upon the context in which the experience is occurring. And then I'll talk about allostasis. What is allostasis? A concept that Bruce McEwen developed in terms of factors that occur during development that change the way our body systems, our physiology work, that is changing homeostatic systems. This, I believe, is really important and an understudied area, because if you look at risk that's connected to adverse childhood experiences, we have this unexplained phenomena that most of the risk, we tend to focus as brain scientists and behavioral scientists on what occurs in the brain. But there's a body that the brain is attached to, or the body, or the body is attached to the brain. But these are integrated systems, all of which are affected by these early experiences. And then I'll talk about a little bit of translation, what we're trying to do in terms of identifying measures that eventually will be scalable in clinical practices. So this is the distribution of the continuum of human sociability. So raise your hand if you're on the very limited, aloof, introverted side of things. Raise your hand. Really? Nobody? This is a skewed sample population. Oh, there we go. I'm not talking about in the context of your academic life, but I'm talking about when you go to a party, do you enjoy talking to strangers, or do you tend to find a corner, right? And how many of you are in the indiscriminate sociability? Okay, so everybody's here? Okay.
Everybody's above average.
Everybody's above average, okay. So we could study cats and dogs. Cats which have limited sociability from my perspective, and dogs that are completely indiscriminate in terms of their sociability. But instead, we've done work now using these recombinant inbred strains of mice that are founded from two different lines. This is C57 Black 6 and DBA. This is the most common strain that's used in genetic studies in mice for reasons that escape me, except that it's historical. It turns out that if you're interested in social behavior, it's actually not the background strain that you'd want to select, and I'll show you why in a minute. So essentially, this recombinant inbred strain, and there are others now that were developed 25 plus years ago, where you basically mated them, get F1 generation, and then made brothers and sisters. So you create a lineage, in essence, in which the genes are either from one lineage or another, the B lineage or the D lineage, and if you have variation in your phenotype that you're measuring, you can actually map genetic loci that are responsible for that heterogeneity. So what does this look like? So if you look at direct social interaction, partner sniffing, this, by the way, is just a continuum of measure of time spent sniffing in direct social interaction, which is an ethologically relevant behavior that rodents perform. This is where Black 6 mice are, by the way, so they're very low on the sociability scale. There are hundreds of studies that delete genes that we think are involved in human disorders, in which we try to reduce sociability, and we're using Black 6 mice for the most part. And of course, they already are terrible at sociability, which is why the results that we have in model systems are not very good. But there's about a five-fold range, 500% difference in sociability in just this one task. If you look at what happens during the social task, the adult with the juvenile, for example, there's about 1,000-fold difference in vocalization during that time. Heritability here, this is pretty close to what Epstein shows for prosocial behavior. He's also showed that the heritability of trust is about 0.2, which means that there's probably not a single gene that's responsible for the development of trust between human beings, which is quite interesting. There was a kid in my third grade class who was like this. Indiscriminate, followed you around no matter what, couldn't take a social cue worth a lick. Right? So this is a direct social interaction. It turns out that that they vocalize during that time. So you can actually break down the measures. I'm not going to show you the principal component analysis that we did. It's published. You can go to this paper that came out in 2018. But you can map these loci. The ones that are above the second line are really highly significant. So for ultrasonic vocalization, we're already working on the genes that we think are responsible for that 1,000-fold range in vocalization during the social interaction task. And there are others that are showing modest associations that we're beginning to map as well to try to understand what heritable components are driving that difference in a prosocial behavior. Now, what's interesting is that you can actually break down that vocalization, which is cued by social engagement, and you can put the animals in different social tasks. So for example, in this task, in this case, on this axis, you have the adult male interacting with the juvenile, so the vocalization is only coming from the male. And you can compare that to vocalizations that are occurring in a different social context. This is a male with female urine and the vocalization response. So you can see in this in this Pearson's correlation matrix that there's probably about a third of the vocalizations using this machine learning software that we developed with engineers at University of Southern California. You can identify syllable repertoires that are different. So this is looking at vocalizations communication. I want to make sure it's very clear that this is not mouse language. This is not mouse speech. This is vocalizations. But you can also change tasks. This is a male and a female together compared to a male with female urine. And in fact, even in this situation, the differences are apparent based on analyzing the syllable repertoires, which are quite complex. So we believe that those vocalizations actually can be broken down to look at the heritable and also the non-heritable factors, and that these vocalizations in social contexts are changing. Difficult to measure before we developed this software, which we again published in 2017 with the engineers who we collaborated with. The other variable, the second variable I just wanted to mention, I'm not going to show any of the data because it's been published, is that there are genes that regulate the rate at which synapses mature. The rate at which synapses mature defines, in some sense, the periods of time during which the circuits are most sensitive to experience coming in. So that's what we define as a sensitive or a critical period. We realize now that the closure of critical periods really is not an accurate representation of what's occurring in terms of the development of the circuits, but it's clear that there are periods of time in which there's greater sensitivity. And so for several of these genes, SYNGAP1, CMET, which is a receptor tyrosine kinase that we work on, if you eliminate those genes, you actually shift the critical period to an earlier period of time. So it starts at the same time, but it ends earlier. So the system goes through a more rapid maturation process. When you do that, you actually change, oops, sorry about that. You actually change the response of the same experience in the same period of time, but the system is now more mature, so that leads to different functional outcomes. So for example, we can shift the sensitive period for binocular vision in the rodent model when we delete these genes that affect maturation by showing that the sensitive period for adaptation to monocular deprivation is shifted earlier, and you lose plasticity in the second half of what would be a normal sensitive or critical period of time. In Tolly Barham's paradigm, which I'll talk about in a little bit, when we do that same experiment in 1-week animals in which we've deleted these genes that are responsible for critical period timing, we actually see a response of hippocampal neurons that is better defined as if you did that manipulation in week 2 or week 3 instead of week 1. So it changes the response. We think that's important because the variation, this is just motor milestones, but the variation in developmental timing is really quite remarkable across infants. We don't really understand why, and so this idea that there's a very specific limited period of time across individuals is, we think, certainly wrong, that there's shorter or longer periods over which these are developing, so the same experience is going to have a different outcome depending upon the maturation of the system. The third variable that I mentioned is that outcomes for the same child can vary depending upon context. This is proposed by Ellis and Tom Boyce in 2005 in the context of this concept of the orchid and the dandelion child. And that this concept of outcomes for the same child can vary depending upon context is even true now for childhood asthma. There's been a variant that's been identified in an immune receptor that the same variant, if the child is exposed to endotoxin in the first year, confers resilience and reduced risk for early onset asthma. If that child is not exposed to endotoxin in the first year of life, their risk for asthma actually goes up about 2.5-fold with the same variant. So it's the same genetic variant, depending upon context, either conferring resilience or conferring greater vulnerability than if you didn't have that variant. In this case, Tom and his collaborators did a study where they looked at pro-social behavior. This, I think, is in 2nd or 3rd graders. This is in the United States. And they looked at children and they categorized them into low court reactivity, the dotted line, and high court reactivity. And then they classified whether the children were in low adversity environments or whether they were in high adversity environments. If you look at the children who show low court reactivity, they tend to show no difference, or statistically no difference in pro-social behavior, irrespective of what their environment is like, what their family environment is like. But if you take the child who shows high stress or high court reactivity and look at whether they're in low adversity or high adversity environments in terms of their family life, you can see that the child with high court reactivity in a low adversity environment actually does better. They've actually extended this to look at other domains of behavior, showing that the same child in supportive systems does actually better than those who show low stress responses, but they fall apart, they do worse in a different context where there's high adversity. So that's another variable. There you go. So G by E, not versus E, G by E, G interacting with E is actually quite complex. And when you look at I was sitting at a meeting one time listening to somebody give the most depressing talk that I had heard, because they kept identifying components of the environment that could drive increased risk for early adverse experiences. This is a list that's relevant to what's occurring in the United States, which is getting more exacerbated, some of these. And compared to the genome, and environmental factors are almost infinite in terms of the combinations that can affect the outcomes of the child. And those experiences, which I denote here as variable 4, is actually the response of cellular and physiological challenges, what Bruce McEwen defined as developmental allostasis, what he thought of at the time of this chemophysiological wear and tear on the system. And now there are a number of examples where this seems to be the case. So the idea is that during development, this homeostatic system is affected, whatever the system is, whether it's response to cortisol whether it's energy production, which I'm going to get into in a minute, or whatever the physiological function is, there's a range. Whether it's sodium channel function or potassium channel function, there's a physiological range. That range changes because of the developmental experiences, and over time, allostatic load creates these prolonged shifts, and these shifts are maladaptive, so over time, they're maintained as the new homeostatic set range and puts, therefore, the system, whatever the system is, whatever you're measuring, in a different state, what some people think of as constant overdrive, that eventually fails. And that may be the emergence of why there's increased risk for all sorts of terrible health outcomes with adverse childhood experiences. So there's Tali. That's you. I decided to show you instead of the model system that we use because you already talked about it. So what are the developmental adaptations to what we would define in this model system that Tali talked about this morning as allostatic load? I just wanted to mention that there is another variable. If you look at C57 black 6 mice in Tali's model, they show these dramatic differences. This is just on and off nest time, how many times they went on and off the nest, and Tali talked about that this morning, that creates unpredictability, and that's really the source of the disruption of the systems that she talked about and that many of us are using in terms of this model because it's so reproducible. And oftentimes in doing science, when you find a model that somebody else developed and it's reproducible, one, you give them a lot of credit, and two, you just continue to use it because it's reproducible. But if you look at DBA, that's a different strain of mouse. They show almost no response. We've done this over and over and over again. They show no changes in nesting behavior in that. So we don't know what the genetic explanation is for this, what the explanation is, but we're interested in trying to understand, using genetic reference panels of mice, if we can identify the genetic loci that are actually responsible for differences in the maternal component of this process, because after all, it's not just how the pups are responding, it's the maternal behavior that is driving it as well. Okay, so here's the model. We use the same thing, 2 to 9 days postnatal, and then we do a number of different measures. And the one that we started with, this is Kathy Eagleson, who's been a longtime collaborator. This is actually California, not Sydney, Australia, where she's from, but we figured she's Australian, we could fool her as long as there was enough beer. Anyway, there had been no work at the protein level to try to understand what the adaptive changes or what the potential allostatic changes were in the system. And so we focused on the hippocampus, which is here, and we worked with a group at Vanderbilt when we first started this that did comparative proteomics. You basically put a single Dalton onto different protein extracts from male controls and male exposed to the limited bedding model, and then we looked at females as well. That turned out to be extremely important. I'll show you why in a minute. So there's just a single Dalton difference between each of these groups, and you can do mass spectrometry, and you can then look and see what proteins go up, what proteins go down. Are there any that change? So you do this by mixing them, and then you run mass spec, and you get these peaks, and you can figure out which proteins are changing by identifying these peptides. So which proteins change? Which class of proteins show the most dramatic changes? Does anybody want to guess? I have another 20 here. Okay. So what changes are proteins that are involved in generating energy. That's in the hippocampus, right? So these are mitochondrial proteins, which are essential for generating ATP. When we first got this data, my shoulders sort of slumped. I thought, oh, non-specific. But it turns out that it's highly specific, that we repeated this over and over again, that there's something fundamental and foundational about the disruption of maternal infant care that's generating a core change in a principal component that's responsible for the ability of a cell, irrespective of the organ that it's in, to function within a normal homeostatic range. So it turns out that we weren't so smart in thinking about this because, again, Bruce McEwen and Marty Picard wrote this really wonderful review in Psychosomatic Medicine. I didn't realize there was a journal like that, in which they described that mitochondrial function might be the node or the nexus for this idea, this concept about allostatic load, that these early disruptions of relationships generates a response that changes how cells in different organ systems need to function under those perturbed conditions. And I'll show you why I think that makes sense. They postulated that the immune system and the neural system and other somatic cells actually, this triggers inflammation, changes in blood glucose that get back to the mitochondria and regulate its energy production as well as lipids. And so they postulated that there are circulating allostatic load biomarkers that one could identify. So if you think about the data that's come out from all these epidemiological studies, the Dunedin, New Zealand study, the Scottish study, the US studies, there are studies all over the world that basically show the same thing. That is, 4 or more ACEs increase risks. So if you notice, none of these increased risks for diseases are brain disorders that are listed. There is increased risk for certain mental health issues, and Tali mentioned them this morning, PTSD, depression, and others. But how do you explain this? How do you explain increased risk for cancer, digestive diseases? I like this category, any disease. How do you explain that, right? And no one's been able to explain why it seems to increase for cardiovascular disease, stroke, 6-fold increase in stroke risk. And if you think about it, if you're perturbing the core or the principal, if you did principal component analysis, what would be the major factor that's probably generating the major variance in physiological homeostatic states? And that's the ability to generate energy to to perform functions like inserting channels or receptors into membranes, for a cell to be able to respond to an external stimulus, et cetera, et cetera. And so we looked at mitochondrial function in isolated mitochondria from the hippocampus in the model system. And so first, because I'm a developmental biologist, I said, well, we need to isolate mitochondria from the developing hippocampus. And I'm not going to go through the assay that we use, but you can measure oxygen consumption. Female is in red, male is in blue. And open circles are the limited bedding condition. Closed circles are control, and you can see that there's an increase in oxygen consumption developmentally. This is just about the time of weaning, so it's pre-pubertal, right? And the males, and we've done these are all individual animals, so we have a large sample size. The male mice don't show any change at all, so it's female specific during development. In the liver, we see no changes in this model. So it's not a non-specific change. It seems to be specific to the hippocampus. We're looking at other brain areas as well. If you look in the adult, we saw a completely different picture, and we were really quite surprised. This idea of allostatic load suggested that there was this developmental response that essentially put the system on trajectory to end up lower in terms of its capacity, whatever that lower means. Here in the adult, in the hippocampus, isolated mitochondria, oxygen consumption, in females is actually lower now. Remember, developmentally, it went up about 30-40% in terms of oxygen consumption. That was the initial adaptation to the perturbations in Tali's model of increased unpredictability. Males show no change, and there are changes now in the liver in the adult that appeared after puberty, not there before puberty. So it's as if the system in certain systems, certain organ systems, have the capacity to manage whatever that allostatic load is, but then over time can't deal with it anymore. And there's a wonderful example of this. How much time do I have left? 5 minutes? Really?
No.
Is that right?
We got 20 minutes.
Yeah, I started late. Who do I believe, 5 minutes or 20 minutes? Okay. Where was I? So there was a study that was done in the early 2000s in which the study looked at trying to understand what the causal explanation was for individuals who died of hypertension in their 30s, which is very unusual. So the study looked at the kidney filtration system, the renal system. And my talk is going to preface what's going to happen in the next session, which is talking about the gut and the microbiome. So they looked at this system and they compared it to individuals who died in car accidents or whatever in their 30s. And what they found is that the individuals who died of hypertension in their 30s had half the number of filtration units in their kidneys. Half, 50% decrease in the number of glomeruli. But each glomerulus was twice the size, right? So in this case, the adaptation was for the filtration unit, which were half in number, to expand in size. But the idea is that over time, that's an unsustainable change. That the allostatic load from having fewer filtration units increase in size, the system is essentially going to crash and burn. And that's what we think we're seeing both in the brain and in peripheral organs, that the initial response is to adapt, increase oxygen consumption, increase ATP because of this perturbation, but over time it can't sustain it, and so it crashes, right? So early life stress response can actually show increases in adaptation, but over time this later life adaptation outcome reverses, and we don't know exactly the time in this animal model. I'm not so concerned about the time actually, because I think this is a foundational property that we think we've identified in terms of why we see risk for all these diseases in adults that emerged at that time. But there was a more surprising discovery. This is Senta Georgia, who works on type 2 diabetes at Children's Hospital Los Angeles. And she and Kathy were having a conversation one day, and she was talking about interests she had in trying to understand what stress did to beta cell development in diabetes. She works on the development of beta cells, which are the insulin-producing cells in the pancreas. And so from the same animals in which we were doing hippocampal work, she looked at beta cells, and there was about a 20% decrease in these insulin-producing cells developmentally. This is right after the period in which the mother and the pups have limited resources. They go back into care as usual at postnatal day 9, and there's about a 20% difference in the number of beta cells. So this is affecting, to a modest degree, likely insulin production in those pups. Look what happens in the adult, right? There's a 45% decrease in the number of beta cells. So it's a persistent change. Now understanding, if you think about how this information first emerged, it was the Kaiser study that, in fact, identified an overrepresentation of adverse childhood experiences in the obesity clinic in which most of the patients had type 2 diabetes, right? So this to me is remarkable because it's suggesting that these homeostatic systems are quite vulnerable to this perturbation. 5 minutes? Now it's 5 minutes. I went from 20 minutes to 5 minutes. Okay, all right. So I'm gonna have to move quickly. So, how do we translate this? And so I'm in part of a research group, a consortium that Jack Shonkoff at Harvard Center for the Developing Child started. And really the goal is not to understand mechanism, but to use a variety of different approaches to determine whether we can identify measures in infants that can be used eventually in pediatric practices to identify those who are most vulnerable. Because right now in the States, there's literally no screening at all. And when you talk to pediatricians, the American Academy of Pediatrics has looked at this, they're reluctant to screen using questionnaires and other things because they're too long, they take too much time, they have about 7 or 8 minutes per patient, per child when they're in with the family, so it's a real problem. So here's the group here. It's an interdisciplinary group, speech-language pathologists, developmental psychologists, pediatricians, and so identify a biomarker panel of risk and resilience for use at well baby visits. The selected biomarkers, Michael Meany is part of this group they're doing epigenetics and other sorts of markers. I'll show you what we focus on with Chuck Nelson at Boston Children's Hospital. This is the study. I show this because I'm a basic scientist. We started doing some clinical research and still do research on children with autism and gastrointestinal disorders, but I wasn't trained as a psychologist, and you can probably tell, and I wasn't trained to do clinical research, and you can probably tell. So this was really hard. 100% of our families in Los Angeles are Hispanic. The mean income for family is about $18,000 a year. U.S. Currency in Los Angeles, that's not a lot of money, right? And so here are some of the measures that we've been doing. EEG, you could argue, is not a scalable measure, but that's what Chuck Nelson does and decided we have to know something about what's going on in the brain, so we use high-density EEG. And we've added more time points. So every infant is coming in at 2 months. We start our measures there and go all the way through to 36 months. And so, here we go. So we've measured using high-density recording correlations between mother's perceived stress. This is in 2-month-old infants, and both beta and gamma power. So high-frequency power is decreased with increased perceived stress on the part of the mom. That's at 2 months. And we have measures now all the way out. We finished all the infants. It's over 100 at 12 months that are showing the same patterns. We're doing wireless eye tracking with Laurent Eady at USC, who does machine learning, looking at natural visual scenes. That's a 2-month-old there. You can actually track a 2-month-old. If you get rid of their chubby cheeks, it's actually a little easier to do, but we can track what they're doing in free-form scenes pretty accurately. And the correlation that we have now that we've just identified is that both in terms of fixation counts and the mean duration of the fixation, there's a correlation between those values and the levels of oxidative stress that we've measured in the infant. So attention and fixation is correlated with status of oxidative stress in those infants over the 12-month period, 4 visits that we've looked at. So I'm going to end here. I didn't talk a lot about social behavior. I did a little bit, but I want to point out that if you look at the key ingredients that programs are developing, one of the key ingredients for buffering early adverse experiences are supportive and responsive relationships, and that makes sense based on what we've heard about today. I'm just going to go to this last one. If you always do what you always did, you'll always get what you always got. So we're trying to change things up. Thank you very much. Okay.
One question.
Uh-oh. I gave you a lot of love.
Love coming back. It was fascinating. It's really fascinating. Now, the question I have is like, there is now data showing, for example, that 700 calories diet, I don't know who would go with this type of diet, can reverse diabetes type 2. So I wonder if you know, or you're doing, or there is any effort to look for, like outside sensitive period, like things that can counterbalance for this risk. I don't know.
Yeah, so I mean, I,
Diet would be compassion and mindfulness would be, I don't know, meaningful relationship.
Well-
What are the factors that in the adult brain counter?
so the answer is we don't know. We've actually reversed the oxygen consumption problem with treatment with Mito-Q, which reduces oxidative stress. It actually reverses it. And so that's an interesting experiment that we did. I think the thing that's going to be more telling in our infant work is the metabolomics work that we're going to do. So I was really interested in the islet cell finding that we have in mice, and I don't know if that's going to translate to human or not. But I was talking to my pediatrician friends. I said, so you guys measure glucose in infants, right? No, nobody measures glucose in infants, right? Nobody. And so I said, well, why don't you? He said, why would we want to do that? And I said, well, what if it was a way of determining that there was some allostatic issue going on? So, what's that?
20% loss of beta cells will not change.
That's correct. So 20% beta cell loss in a mouse. I don't know what that translates into human, but we think that it'll be important if this is correct, if the animal model work is correct, that there are fundamental metabolic changes, which I really believe are occurring, and Tali's shaking her head yes, that's really a component that we have to push on pretty hard, because I think that's the underlying root cause for why we see wide-ranging increases in disease risk across organ systems. It's hard to explain any other way. Okay, coffee break.